Hello, my name is Piazzon, and I'm going to present the results from a study where we determined the effect of genetic selection for growth in intestinal microbiota and disease resistance in GILTHEAD seabream. This work has been performed by the Nutri group and the fish pathology groups at the Institute of Aquaculture of Torre la Sal in Spain and is part of the European project AquaIMPACT. So, genetic selection for economically important traits has been applied successfully in GILTHEAD seabream aquaculture. In particular, we have been working with some families from a previous Spanish project that had been selected for heritable growth. So, we had a group of families that were selected for fast growth and a group of intermediate and slow growth families. Our previous studies demonstrated that the fast growth families showed a better growth across seasons, and they were able to reshape their intestines when fed plant-based diets in order to maximize nutrient absorption with no differences in growth. The intestinal plasticity that these families presented was genetically regulated and correlated with changes in liver and fat metabolism. So this way, fast growth families were able to adapt better to alternative diets with no impacts on growth. The next step, then, for us was to determine the role of the microbiota in these different families. Intestinal microbiota is not only important in nutrient absorption and metabolism, but it's also key in immune system training and also in disease resistance. So that's why there's a lot of interest in manipulating this microbiota to improve the health of the animals in production. However, this is not an easy task because intestinal microbiota is composed by very complex communities of microorganisms that can be affected by many different factors, among which, and for the purpose of this study, I will highlight that microbiota is very affected by changes in diet and also by the genetic background of the host. So, in this study, we wanted to know if the genetic background of GILTHEAD seabream is influencing the intestinal microbiota. Also, how these bacterial communities were shaped when the different families were fed alternative diets and how the different families responded to intestinal disease. So, in all, our aim was to study the interaction between the selection for heritable growth and the metagenome and how this interaction affects diet plasticity and disease susceptibility. So for the experimental design, we use fish from three different groups of families: the fast, the intermediate, and the slow growth families. All these animals were tagged and kept together in the same tanks, and they were fed during 9 months with a control or a well-balanced plant-based diet. After the 9 months, we collected the mucus from the anterior intestine to extract DNA and perform Illumina sequencing to study the autochthonous bacterial communities of the different groups. We used anterior intestine because this is a part of the intestine where nutrient absorption mainly takes place, and we are interested in that. And we studied the adherent or the autochthonous bacteria because these are actually the bacteria that are living and staying within the host. So they are able to interact more tightly with the macroscopic host, and probably they have more effects at a metabolic level. In parallel, we also perform an experimental infection. So for this, we use two groups of families, the fast and the slow growth families. Again, the animals were tagged and kept together in the same tanks. And in this case, all the animals were fed the plant-based diet because we know from previous studies that this diet can slightly increase the susceptibility of the animals to get infected with the parasite. And in this case, we wanted all the animals to get infected, so we used the plant-based diet. We infected the animals with an intestinal parasite that's called Enteromyxum leyi. So this is a parasite that lives and divides between the enterocytes, causing epithelial disruption and all the problems that are associated with that. We waited for 70 days for the infection to develop, and then we performed parasite diagnosis by qPCR. Moving on to the results, we found that there were no differences in richness, diversity, or composition at the phylum level among the different groups of families. So all families presented the typical microbiota of GILTHEAD SEABREAM composed by around 90% of Proteobacteria, Firmicutes, Actinobacteria, and Bacteroidetes. But looking at the effect of the different diets, we could find important differences at lower taxonomic levels. So this is very easily seen in these discriminant analysis results. So here we can see the fish in the three different families, the fish that were fed the plant-based diets in red and the fish that were fed the control diets in black. And it's very clear that the change in diet induced a change in the microbiota in the different groups of families. But when checking at the statistics of these models, we can see that these changes are significant in the slow and in the intermediate growth families, but they are not significant in the fast growth families. Studying into more detail the bacteria, the OTUs that were changing with the change of diet, we could see that most of these bacteria changing were actually family exclusive. So this means that the shift in microbiota driven by diet are genotype dependent. Also, checking the relative proportion that this changing bacteria constituted relative to the overall bacteria present in the intestines, we see that the change in diet was inducing a change of around 30% of the total bacteria in the intermediate and slow growth families, whereas only 9% of the bacteria were changing in the fast growth families. In an attempt to evaluate what these changes of bacteria induced by the diet were meaning, we perform inferred metagenome of these bacteria that were changing and perform a pathway analysis. Interestingly, these 9% of bacteria changing in the fast growth families when fed alternative diets potentially accounted for changes in around 17 metabolic pathways, whereas only 11 and 4 pathways were changing in the intermediate and in the slow growth families. So this means that the small changes detected in the fast growth families were enough to significantly change the metabolic potential of these populations, while this was not so efficient in the intermediate or in the slow growth families. Well, of course, we don't have time to go into detail on the bacteria or the pathways changing here. There's a lot of information, but just as an overview, I can say that the changes happening in the fast growth families when fed plant-based diets were in the order to produce an anti-inflammatory profile in these intestines, while the opposite inflammation was observed when slow growth families were fed the plant-based diets. Finally, here we can see the results from experimental infection. Here we see the growth of the animals. So as expected, the non-infected group, the control group, the fast growth families grew more during the 70 days than the slow growth families. But when infected, the weight loss, which is one of the most evident signs of this parasite, was lower in the fast growth families. And this correlated with a lower parasite intensity and a lower parasite abundance in the fast growth families. So to conclude, we saw that genetic selection for heritable growth influences intestinal bacteria in GILTHEAD SEABREAM. There's a clear correlation between genetic background, microbiota, and dietary and physiological plasticity. These results, together with our previous findings, show us that the GILTHEAD SEABREAM families selected for heritable growth are very robust. They are able to adapt better to diet changes, reshaping their intestine and organosomatic indexes. They can cope better with pathogens, and they harbor a plastic microbiota able to efficiently adapt to changes. The next steps we are conducting now are a metatranscriptomic analysis of these bacteria changing to validate the inferred pathways results. We are also working trying to find the mechanisms that correlate microbes' health, growth, and disease resistance. And we are performing a dynamic profiling of the microbiota through the whole production cycle of selected families. Well, of course, I only have time to go through the highlights of this research, but if you want more details, all these results have been recently published in Microbiome. And you can find the reference here. There's also a nice video with the summary of the results. And that's it from my side. Thank you very much for your attention, and I will be around if you have any questions. So thank you.